I should be thankful to the organizing committee to invite me for this talk. And my talk will be on the generated immune response in COVID-19 and therapeutics and diagnostics. Let's go to the second slide. This pandemic is ongoing, and WHO declared it as a health emergency on 30th of January and confirmed as a pandemic on 11th March. 229 countries are affected by the pandemic so far, and total confirmed cases are 628 million, and total number of deaths are reported 6.5 million. So the effective way of transmission is through the contaminated air droplets from the infected patients. And historically, we had 4 coronavirus outbreaks in the ancient times. They are still exist, but in the mild form. However, in the past 2 decades, we have 3 outbreaks reported due to coronavirus. First was in 2003 with SARS-CoV-1, and in 2012, it's with the MERS virus. And now we are facing the new pandemic with the novel virus SARS-CoV-2. So the common symptoms of the virus are cold, cough, fatigue, shortness, and loss of smell, etc. And people have symptoms like from asymptomatics to the non-severe symptomatic stage, and the finally severe symptomatic stage. They can, the symptoms appear at various levels. That is the most difficulty to deal with this virus. And the immune response is regulated by TLRs and other receptors. And the most pathogenic developed form by this virus is the cytokine storm formation, which starts forming in the lungs, and it can be disseminated to the various organs. So the pathogenicity of this virus is, what are the outcomes of pathogenic outcomes? We normally do not know how the asymptomatic patients, they transmit the virus and how they have the ability to eliminate the virus. If they have the ability to eliminate the virus, how come they transmit? And we have seen that throughout the pandemic. It's all the progression of the disease. It depends upon our overall immunity, and this virus plays to lower down the immunity as well. And initially, in the initial stages, the antisera and the pegylated IFNs, they use strategically to boost the protective immune response. So initial convalescent sera was used as well. Monoclonal antibodies are now available because they work more specifically. People have used the mesenchymal cell therapy too, but it didn't work at that stage. It is still ongoing. Research is going on this mesenchymal cell therapy. So let's go to the next slide. This is shown as the how virus infect the cells, and it goes through the AC2 receptors, and basically the macrophages and neutrophils, they have impaired activity when they are getting infected with the virus and they produce the pro-inflammatory cytokines. On the other hand, APC cells, they generate the immune response and they activate the T and B cells. So now how the immune lapse of SARS-CoV-2 takes place. One is impaired immunity, another one is the high viral loads. So the high production of the tissue factors, first is the coagulation cascade takes place. And in the macrophages, instead of these, normally myeloid differentiation 88, this adapter, and there is a tissue necrotic factor adapters, they got activated normally. But these pathways got impaired and P38 pathway get activated and they produce the chemokines and cytokines, and especially the pro-inflammatory ones. And after that, these pro-inflammatory cytokines attract more macrophages and they got excited and they settle down at one place. And VEGF, a growth factor, which can cause the hypoxia condition, and ARDS established. And patient can die here. But as the cytokine storm, it has ability to pass through the other organs through various channels, and it results into the multi-organ failure as well. So, and it ultimately leads to the death of the patient. So that's how virus triggered the immune response and result into the fatality. So let's see the next picture here. This is the SARS-CoV-2 infected lungs, and this is the lungs with the other community-associated viruses. And now you can see the difference here. The cytokine genes and ISG genes, there is the dense formation of these activity of IFN, it just goes totally impaired. And as a result, these cytokines establish the pathogenic reactions. And here's the picture of bronchioles. And here again, on the one side, it has shown that how it forms the cytokine storm here. And the oxygen is stopped going through the lungs to the body. And here it activates the VEGF. And the hypoxia condition can be produced due to the accumulation of the cells. And one more thing I must add is there's a high uranidase synthase enzyme gets activated as well. So this is the actually area we look for making the therapeutics. So the next slide is, so in this pandemic, most of the drugs were repositioned, which were trialed in the past, and doctors, they have the previous experience with those drugs. But some of the drugs, they were on the trials as well, the new drugs as monoclonal antibodies. So I will try from step to step. The first is used as the chloroquine and hydroxychloroquine. This is the most controversial drug, I must say, because in USA, in most of the patients, this drug was found to be responsible for increasing the heart rhythms. But the WHO has given this hydroxychloroquine regime as 1 to 3 doses on the first days and 1 to 2 doses from 5 to 21 days. Especially this hydroxychloroquine, it works well for the people in the eastern countries. The next is the lopinavir and ritonavir. This is actually the drug used against HIV, and it was used with IFN in most of the countries. And tocilizumab and cerulizumab, these are the monoclonal, especially to reduce the activity of the interleukin 6, which is the most activated form of protein, cause the hyperinflammation. The dexamethasone and corticosteroid. These are the anti-inflammatory drugs, and they reduce the death by 30%. Favipiravir and remdesivir. They are both inhibit the activity of RNA polymerase, RDRP. So yeah, the next is the ivermectin. Ivermectin is not the antiviral, it's antiparasitic. And WHO doesn't recommend this drug until unless it is used in context with any clinical trial. Nifamostat, chemostat, these are the antagonistic to serine protease. And there are famotidine and arbitol. They have again worked well, and they block the fusion of the viral and cellular membrane. So these basically drugs, they are mainly chosen to stop the invasion of the virus and work on the proteins of the virus like protease and RNA polymerase. So they reduce the chances of death of patients who are prone to develop the hyperinflammatory response against the virus. So this picture is showing that the chemostat and nifamostat is working against TMP or SSA2. So here it stops the invasion. Chloroquine, hydroxychloroquine, azithromycin, it reduces the protein formation because it increases the pH inside the endosome 1,000 times more than the natural pH increase. So obviously it decreases the virus replication. Remdesivir, favipiravir. Remdesivir used in USA and favipiravir used in Japan. They work against the RDRP, RNA polymerase, RNA dependent and RNA polymerase enzyme, hence reduce the activity of virus. So there are other monoclonals like reagent monoclonals. It has been used, but it was not found to be reactive against all the variants. Bamlanivimab, these monoclonals are revoked by AUM. But then it outcompeted the Delta and reduced the risk of hospitalization by 70%. So molnupiravir, it also interferes with the RDRP, and Paxlovid, it disrupts the 3CL protease. So Paxlovid is quite famous now. So I'm actually giving the brand names of these drugs, but normally these drugs are made to deal with immune system and to stop the virus replication. Convalescent plasma, it was used initially, but WHO lately recommends against this plasma because it wasn't working well in most of the patients because of the continuous development of the new variants. Synthetic interferon, they don't work well as well. So NIH doesn't recommend interferon therapy. So let's go to another slide. So these are, I have described the drugs for repurposing. And now I'm going to explain about the vaccines because vaccines are the effective tool which develop the active immunity in the normal population against the virus. So there are different vaccine platforms developed. One is the viral vector vaccines, excuse me, and DNA vaccines, RNA vaccines, live attenuated vaccine, and protein-based vaccine. And I must explain here, these all vaccines are made on the new platforms. We are not in the old era because as we have seen during HIV infection, the infection continues instead of having other medicines. And obviously, these all viruses, like coronavirus, Ebola virus, they are the new emerging viruses. And we obviously need those platforms which are actively work against the virus instead of producing any adverse reaction. So these all vaccines are made on the new platforms. So the viral vector vaccines here, the viral adeno-associated viral vectors are used, and the sequences of the protein, surface protein of the virus, it goes to the nucleus, and then from the nucleus, it's translated into the surface antigen protein, and which in turn produce the immune response, develop in the development of the antibodies against the protein. And the DNA vaccines, they are plasmid vaccines. They again works the same way, goes to the nucleus, and then from nucleus, it's translated into the surface antigen. So the next is the RNA vaccine, messenger RNA vaccine. These are the most famous vaccines now because of their lot of benefits. And the new vaccines, the biggest advantage of this next generation vaccine is they can be produced in the small footprint facility. The RNA vaccine, again, it just works directly outside the nucleus. It doesn't go inside the nucleus, so we will not be having any issues regarding epigenetic transformation, etc. So here, its RNA is translated the sequence into the surface protein, and ultimately it produces the response, innate and adaptive immune response. The live attenuated vaccines, They are, again, they are not produced in the old ways, like in the older time, we used to have besieging the vaccines at some stage where we see the virus goes avirulent, and we use that virus or bacterial strain for as a vaccine strain. But it's different. It's a synthetic form of the virus where we work on the open reading frames and um, make its form as a not infective. It's not a virulent form. So it just goes into the cytoplasm, replicated it, and produce the immune response. Now, the last vaccines are the protein-based subunit vaccines. They are again synthetically produced, and they work just outside as the virus. This antigen response is produced by the cells, and they develop the immunity against it. So it's amazingly, if you see, our cells are so smart and intelligent. They work in every respect. These all platforms they have different mechanisms, but our cells, they have the capability to produce the immune response. Now, this is a picture of mRNA vaccine. It's how it works. It is wrapped with a nanoparticle, lipid nanoparticle, and as it administered, it produces the spike protein, and these spike proteins are translated separately, and they combine in the cell, and they make the active form of spike protein inside the cell. So we don't need to use those olfactories to establish in vitro or outside just to put the response. It's a very smart way of utilizing the human cells in vivo. And through MHC, it produced the response. So the B cells generate the antibodies to neutralize the virus, and T cells start to kill the infected cells as well. So how the immune response is produced. So here, the next is how the innate immune response signaling takes place. These are the different forms of the RNAs. And here, the signals produced through myeloid D88 signaling pathway, RIG, MDA. And these pathways ended up here, IRF, this is the interferon factors to the nuclear factor. They activate it, and they activate the formation of interferon here, and this signal goes from the JAK-STAT pathway, and they are the receptors. So as the interferon produced here, if they are regulated, they work in the regulated form, and this happens generally when in the seroconversion stages, and ultimately they develop the adaptive immune response as well. So there are so many variants produced by SARS-CoV-2, but the most famous are the alpha, beta, gamma, delta. They have done a lot of devastation around, and the Omicron is going on since 2021, and it has so many subvariants which are still getting activated and evading the immune response. So variant of concerns are the variants which generally lapse the immune response, and result in forming the breakthrough infection. So it is very important to combat with these variants is to update the vaccines. And obviously, if there is, obviously in the hotspot areas, they need to put the lockdown to reduce the transmission of the virus, because the variants have specifically this quality that they have um, they have skills of active transmission and producing more infections. So let's come to the diagnostics now. So diagnostics are mainly is the RT-PCR method used, and this method was used initially in the previous SARS outbreaks as well. So in this method, yeah, and the second one is the LAMP method, and CRISPR-Cas9 method has also been established, and the immunodiagnostic approach, serological test, and the next generation sequencing. So in RT-PCR, the RNA is extracted and it is transcribed into the complementary DNA. So the primers are attached with the probe and it releases the dye. And it is, if there is sequence is specific, it works well. And so threshold cycle is increased, and this happened in the positive patients. So there are a lot of advancement made in the RNA extraction with the nucleic acid amplification techniques. These are the RT-PCR method, nested PCR, RT-LAMP method, RT-PCR method, gene spurt method, and magnetic nanoparticle based method. So the LAMP amplification initiation assay, here we use the multiple primers, forward and the backward primers, to initiate the cycle. And it's the isothermal, so we don't need to have various temperature variations in this method. That is the advantage of this method. However, there are too many primers, so this is the difficult situation to use this method as the standard gold method. So next method is the CRISPR-based Cas39 method. And in this method, RNA polymerase is used to convert RNA, extracted RNA into DNA, and T7 transcription and C13A protein is used, and it can be used as a Sherlock method, in the Sherlock method directly as a Cas39. And the best part of using this test is that this test can be conducted in one tube, and the test can be produced into the strips. However, the sensitivity of this method is not up to the mark at this stage, but this is the most economical method if it is established in the epidemiological area, and it can be used in those laboratories which are in the remote area and they may not have those sophisticated instruments to detect the infection. So this method can be used. So there are immunodiagnostic methods, other methods like rapid antigen detection method. This has been widely used in the population. And in this method, again, the antigen virus is detected through the conjugated level antibodies, and these antibodies are reacted with the primary antibodies, and ultimately the complex is detected by the developer. So now, normally IgM antibodies are developed before the IgG has to develop. However, there is a one proposal as well. If IgA can be detected with IgM and IgG, it can make the difference, because when we detect the antibodies, they can tell us that there was an infection in the past, or maybe ongoing if antigen is there, and it doesn't apply for the immunized people, because obviously immunized people, they develop the antibodies. So the next is the NGS virus genome amplifications method. Here the cDNA synthesis takes place, and library preparation is done, and the sequencing is done which is aligned with the reference, and these sequences are used for the phylogenetic analysis. That is why how we are recording many variants. So the interpretation of the diagnostics are mainly based upon the situation of the patients. That its interpretation depend upon the different circumstances. So this table shows the clinical interpretation based on the review summary on the RT-PCR and antibody test. If they all are positive, then obviously it determines the acute infection. If the RT-PCR is negative and the others are positive, it can be recent infection or late onset infection. Similarly, if they all are negative, then there's no infection. So there is another new emerging fact developed, that the gut microbiome gets altered during the infection, because they first found in one of the studies happened in Italy that they have noticed that the hospitalized patients, they reported as the enterococcal septicemia. So they have determined their fecal microbiological analysis, and they found the bacteroideaceae, leptospiraceae, and the rheumaticocalaceae. These families were depleted, but the pathogens like enterococcus, staphylococcus, and streptococcus, serratia, etc., they were getting developed. So obviously, there was an impaired immune dysfunction was reported. So it normally happens when there is a clear transportation of the metabolites and pathogens from the gut to the lungs takes place. So nowadays, people are using probiotics to see their actual effect in COVID-19 patients, but there are some studies done in the past as well, taking the lactobacillus, and it was known that they increase the IL-10, interleukin 10 and 4, and decrease the IFN and tumor necrotic factors. So obviously there is, obviously there is some system that probiotics may involve to improve the immune system in the bodies, because they have capability to produce SCFs, and these SCF fatty acids, they work in different ways. They immunomodulate the cells and the gut environment, and which reduce the invasion of the SARS virus. And I haven't written here, there is one more study I have seen that people have found the sequences of the virus inside the probiotic, inside the microbiota as well. So it again gave us the clue to have more studies to be done, especially on the alteration of gut microbiota. So the final remarks are the increasing the risk of spreading the multiple variants of concern provide the strong indication to produce the multivariant vaccines, that is the pan-variant vaccines we need to work effectively against all the variants. It's hard to predict which variant is emerging and which is becoming the dominant to lead the pandemic. So in order to control the pandemic, there should be some provision should be made, like COVAX and African Vaccine Acquisition Trust. They deliver the vaccines to those areas where we don't have provision to make the vaccines. So it is imperative to have the knowledge of the emerging disease pathogens and design new pandemic preparedness programs in context with the strategies to conduct the research on the potential vaccine candidates, therapeutics, and rapid diagnostics to combat the infections. It could reduce the morbidity and mortality. So thank you very much. This is the final remarks, and I tried my best to explain everything. Yeah, so in this presentation, if I see there are tremendous opportunities for the researchers to pursue the research on different topics. If you see as such working on vaccines or any therapeutics, it takes years and years. So I have just given the overview of the therapeutics and diagnostics. Thank you so much.